want to introduce Cathal Walsh. I decided I was going to get the title right, so I wouldn't have to look over my shoulder. So, Bayesian approaches to health decisions. Also to congratulate Jamie, who's hiding on the back, who just finished a successful bike.
Well done again, Jamie, and thanks very much for the invitation, Tony. So I'm going to talk about something which you'll see on telly tonight as well. So on Clareburn Live, if you're watching, this topic will be relevant to that. It's very topical at the moment. There's a drug going through assessment, or has gone through assessment, and is being discussed at the moment for cystic fibrosis. So there's publicity around that today as well. But this topic is to do with a grant that we've got a couple of years ago now, and in fact midterm review, a HRB Research Leader Award, looking at the area of health decision science. It's a couple of partners, so the health-related partner is the National Centre for Pharmacoeconomics, that's involved in doing the evaluations of new technologies and drugs for the HSE. The Universities of Sheffield and York, SHAR in Sheffield, and the Centre for Health Economics in York, and Health Decision Science Group at the Harvard School of Public Health in South Boston as well. So collaborators from all those institutions involved in the grant. Well, here's Limerick. The last time I gave this talk or described this work was when I was in Africa a few weeks ago in Tanzania, and they asked me, where is Ireland? And I had to show them a map of Ireland, and in particular they wanted to see Limerick. They hadn't seen anything as green as that. I explained to them as well that Ireland, you know where Guinness comes from. What was the response, do you think? Guinness comes from Nigeria, is what they tell me. So the world is different in different places. But that's my home institution now, UL. The question of decision science then is relevant to statisticians. It's about making decisions, and in particular we're making health decisions in the face of uncertainty, complexity and competing values. Here's a statistician's view of the world, perhaps. We might think in terms of p-values. So a p-value is something that people might be familiar with. We have an idea that it's related to some data, related to a null hypothesis, and maybe some theoretical sampling distribution. But do we really know what it means in terms of decisions? Well, we might have some problems with p. Let's say a scientist does a well-designed experiment, could be a clinician, perhaps clinical trial, and we want to do some inference about mean treatment. And we get a p-value of 0.04. What should we conclude? Well, I'll give you some possible conclusions. We could conclude that the data prove that the treatment works. Or there's a 4% probability that the null hypothesis is true. Or, given the data, there's substantial evidence that there's a non-zero effect. Or, if the average effect is really non-zero and the model is correct, then the data observed are contained within an unusual subset of all possible sets of data that could arise. Only one of those things is true. Which of them is true? The last one is true. So a lot of the other things that we'd like to conclude on the basis of p-values, we can't actually conclude. We can't conclude that it proves the treatment works. We can't say that there's a 4% probability of the null hypothesis being true. And we can't say it's substantial evidence. 0.04 isn't substantial evidence. And that's a challenge that arises when we make decisions about new drugs. That in a clinical trial, if the p-value is less than 0.05, we reject the null hypothesis and we say there's a treatment effect. And then otherwise, we can't claim that there's a treatment effect. And the way in which drugs are licensed is, the decision is, if the p-value is less than 0.05, give it a license. If it's safe, otherwise don't give it a license. And that's problematic from a couple points of view. Well, one is 0.05 might not be a strict enough threshold. And also, if by chance the study wasn't powerful enough, you might get rid of a treatment that could be beneficial. And so our trial was flawed because it was insufficiently powered. But in terms of using the drug, we might think of other things. So let's imagine a situation where we have two treatments, treatment A and treatment B. And an RCT published in the New England Journal of Medicine shows that A works better and the p-value is 0.013. So what else might you like to know as statisticians or clinicians or epidemiologists? What else might you want to know?
Effect.
Yeah, so the effect size. What's the effect size? give us an idea of the effect size, and maybe give us a confidence interval as well. So here I go. The odds ratio is 6.0, and a 95% confidence interval goes from 1.5 to 24.7. Now, which would you use? Well, I mean, it's a leading question. A, right? Because it's better. Maybe. Or you might want to know more information. You might want to know, well, what's this treatment for? Well, let's say treatment A costs €3,000 compared with €15 for treatment B, and you need to take 2 months off work to undergo treatment A. And the condition in question is ingrown toenail, and the outcome is recurrence-free at 1 year. Now, which would you prefer to do? Treatment A or treatment B? Treatment A might be 6 times better, but that inconvenience of 2 months off work might be a substantial reason not to try treatment A first. You might try treatment B first. And also the financial imperative might mean that you prefer to give treatment B a go. The same text, same numbers, but in this case, the condition in question is congenital heart defect, and the outcome is survival after 5 years. In that case, which would you prefer? Well, you might be willing to pay the €3,000 in that case and take the time off work. So there's something in the additional information that makes us change our decision or change our choices. Good time to introduce an old theorem. So this is Bayes' theorem, and it's an alternative to thinking in terms of p-values or in terms of statistics as we might have done. One of the nice features of the paradigm is that everything is probability, and everything that we don't know is treated as a random variable. And so we never condition on something that we don't know. We condition on data, but we don't condition on something we don't know. So a measure for the parameter values of interest given data depends on what we knew before or what we believed before, and something to do with our experiment or likelihood function. And it tells us how to update our beliefs in the face of knowledge. If something is, we could do the same thing as the statistically significant decision, so we could use the same rule, but we can do otherwise, and we can incorporate, as well as probability, something called utility. And the way that utility theory works is straightforward. So here's a situation like the previous one. The square is a decision node, so that's where you get to choose. The circle is a chance node, and that's where God, Tai Chi, something else decides for you. And imagine a situation where you take an ointment. Well, that might have an intermediate value. It's not the perfect solution. But if you undergo surgery, there's 2 possible outcomes, and it's just chance decides. Either it works very well, and you're cured, and that's the best outcome, so utility of 1, or else your toe falls off, and that's the worst outcome, and that's utility 0. And there's a certain probability of success, based perhaps on experience or based on a clinical trial. We can estimate what that probability is. And we get to choose the surgery or the ointment, and then chance gets to decide whether or not we lose our toe or it stays and we're cured. And the values are on the right-hand side, and there are values about how we would rate each of those outcomes. Best, worst, and then something in between. That's how we've rated these ones. Well, the Bayesian decision-making framework is about subjective expected utility, and the argument is that we should maximize subjective expected utility. So considering all courses of action, we choose the course of action which leads to the best expected outcome in terms of our utility. So here's the picture that we had before. We get to choose between treatment A and treatment B, and there are 4 paths, if you like. Treatment A might work or it mightn't work. Treatment B, and it works or it doesn't work. And we consider the utility of the success of treatment A combined with the costs in financial, personal, and in terms of side effects as well. And for the failure of A, success of B, and failure of B. And so we can write that in shorthand like that. Based on the probability of success, conditional on choosing treatment A, and probability of success given that you've chosen treatment B, we can work out the expected utility under each of those paths, and we choose the treatment that maximizes subjective expected utility. And that allows us to distinguish between the situations that we had before. So we've separated out probability and utility, And now we can say that it's okay that if the condition in question is congenital heart defect, I'm willing to take greater risk, perhaps, of side effects, to pay more money, and to take time off work, because the value of the outcome is much greater to me than the value of the other outcome. So utility allows us to distinguish between those situations. Which leads us to the question of health technology assessment, and that's what will be covered this evening, or perhaps what was in the media recently. And it's a structured framework for assessing technologies, be they drugs or devices. And that contrasts with regulatory considerations, which are about whether or not something's fit for purpose. In other words, and in the going off topic here, but in the case of the drug in question, the one question is it safe? Is there adverse safety profile? And if there isn't a safety signal there, the next question is, does it work? And in the case of the drug in question, the company claimed that a 5% improvement in lung function was a clinically meaningful effect. In fact, in their trial, they found a 2% improvement in lung function, which was far smaller than the clinically meaningful effect that they set out to show, but they had powered the trial sufficiently so that they showed a statistically significant effect. So yes, it was a statistically significant effect. It was small. It was only sustained for a moderate period of time, but there was no safety signal. And so the question of giving a license is there. Yes, you can give the drug a license because it meets those criteria. But the other question is, where does the technology fit amid everything else that we want to do for that group and for everyone else as well? Well, here's the utility function, one utility function that we could use. So imagine this is your life. This is a quick view of your life. If you could measure it on a scale of 0 to 1 at any point in time, you have your ups and downs. So you're wandering around through life, ups and downs. You might have an up when you're going out for your Christmas party, and then the next day it might go down a little bit for whatever reason. But then there might be a point in life where we find that there is something happens. So something happens, and let's focus on what happens here. We'll zoom in. So something's happened to this person. So it might be a chronic condition which is progressive, and they become progressively more disabled. And at age 60, then they cease to be. So the utility is 0 from that point on. And these are alternative courses of action. So here we've got a treatment which will improve quality of life, but we see that it doesn't extend life. So the quality of someone's life is better, but you live for the same amount of time. Well, there's an additional benefit, is the area under this curve. Here we've got a treatment which will extend life, but perhaps because there's a risk of side effect, there could be a risk of hemorrhage or stroke or some such, but that the life is extended, but the quality of life is not so good. And so the question is, do you prefer the black line, do nothing? The red line is improve the quality of life for now, but knowing that you won't extend life? Or do you take a chance and try and extend life, but perhaps with a reduced quality of life? And whose choices are they? Well, they're subjective choices, and the answer is the person themselves, perhaps, is the person who should decide on those trajectories. But in the QALY framework, once we have a tariff, in other words, once we know what this utility function is, we maximize the number of QALYs. And in this case, if this was a true reflection of what the likely events were, then it's the red course of action that we'd go down. We'd say we'll maximize the QALYs. The question of how they're obtained is about valuation. We've done a 3-level valuation for a societal group, so in other words, surveying people within society and asking them about trade-offs for different conditions like depression versus mobility. We're asking explicitly how people would trade off between those things. We're also setting out to do a valuation of those with patients as well, and that's a grant that's about to start. And in Galway, they've done a valuation for the 5-level EQ5D. So the EQ5D is a quality of life instrument, 3-level and 5-level, and we're doing patient valuation. Galway have done the societal valuation for the 5-level. Well, once we've got those values, we can plot every intervention on a plane, a cost effectiveness plane. And these seem like simple decisions. Well, if we have a collection of values in the bottom right quadrant there, so improved quality of life gain, and reduced cost compared to what you were going to do, then that's something we should do. Improving quality of life and reduced cost, do it. If we've decreased quality of life and increased cost, then certainly don't do it. And some interventions are in fact in this area. So where the side effects are substantial in the long run, then you may end up in this quadrant here. And this is where we typically end up, which is we've an improved quality of life, but at a cost. And the question is, should we do it? Well, put it on the list of things to do. Improve quality of life, let's do it. Let's do it if we can afford it. So simple decisions, red, green, and orange, or maybe not orange, maybe it should be shades of red and green. And shaded in this fashion here, using something called incremental net benefit, which is the quality of life gain times some parameter lambda minus c, where c is cost. Well, how do we choose lambda is the question that arises here. And the answer is, there's a theoretical answer and there's a pragmatic answer. And the theoretical answer says, well, we'll find some value so that all the stuff we can afford falls under the threshold and the stuff we can't afford is above it. Well, if we do a thought exercise, then we can think of all the possible interventions that we have, and there's a ICER associated, or an incremental net benefit associated with each intervention. And the interventions that are effectively cheap or low down on this plane have low height, and the area is proportional to the total cost. So the utilization along this axis, and then the cost per quality on this axis here, and this is the total cost is the area. Well, we just order the interventions in this fashion, and when we run out of money, we stop paying for things. So we do all the stuff that's good value for money, if you like, the stuff that's relatively cheap for a given gain in quality of life. And when we run out of money, we stop doing stuff. And this is, as far as economists are concerned, the most efficient way of allocating resources. So things that have a very high cost per quality of life gain are things that we end up not doing. Because if we do those, then we don't do the stuff down here. So for every person that gains an intervention here, there might be 10 or 15 people here who do without an intervention. Same way as treatment for a disease, maybe at end of life, for example, could be very expensive, and we do that at the cost of screening people that we otherwise would catch early. Talking about screening, here is an analysis for the colorectal cancer screening program, looking at different technologies, and to bring out in this is the gain for these 3 different technologies on average per person. Some people gain more than others. The people where cancer is detected are the people who gain. The people who are screened and are screened negative and don't have disease don't gain anything, but the average gain is given on this axis here, and the average cost per person is given on this axis here. So we rule out technologies that fall up here by something called extended dominance. In other words, there's a linear combination of these technologies that we could undertake for the same price that would yield greater health benefit. And so, on a societal basis, what we do is we give the majority of people this technology and some people this technology, and the average gain would be much bigger than if we did the red technology here. And these are just two different fecal tests, so looking for occult blood, and then one flexi-sig test. And so the most efficient of these was FIT, and that was what was recommended on a population basis. And the question is, if this line here, if the slope of this line is sufficiently shallow, we recommend it. Otherwise, we might recommend earlier technologies. But if we're thinking in terms of statistics, we don't have a fixed value. What we have is we have inputs to a model for progression of disease. And we place probability distributions on these inputs. And conditional on these probability distributions, feeding them through a model, in fact feeding them through a system, we end up with a distribution on the cost-quality plane. So we induce a probability distribution on the plane. And if we think in terms of Monte Carlo simulation, we'll end up then, instead of points on the plane, we end up with clouds on the plane. And in this case, the FIT technology turned out to be to the right and at a modest level in terms of cost, and to be recommended even though there was substantial uncertainty about the actual benefit, average benefit that would be gained, and the actual cost per person. So we can think of interval estimates on this plane rather than point estimates, but the technology still dominates the other possibilities. So we're bringing uncertainty together in with the cost effectiveness or the HTA models. Well, let's bring in Bayes and HTA together, and so that's bringing this, a lot of people there's still some people in Trinity, which is where I used to work, and people in Limerick involved, as well as people in the NCPE, in order to try and make decisions in a structured way. Well, we might be doing research on information about the costs and effects, or we might be doing research on the utility of the decision maker. And there's three particular areas that we're working in. Two of them I've mentioned already. So getting a tariff for the population for EQ5D, for the 3 level for the population, is something that we've done the field work on already. Doing field work on the trade-off, utility trade-offs for patients is something that's starting from, it's due to be this autumn, but it'll be the start of next year. And the other topic is something else, which is called multi-criteria decision analysis, which is a different framework from the cost per quality framework that we've seen already. So the idea is that we look back at previous decisions that have been made in Ireland, effectively through the political system, because some technologies that you might have classified as not cost effective using the models that we put forward have been granted reimbursement. So the question is, what does that reveal about us as a population? Well, by looking at the various factors associated with these interventions, extract information on those technologies. For example, does the technology involve end of life? So is it an end of life treatment? Were there children involved? Is it effectively a drug for children that we were looking at? Can we classify the quality of evidence as high, medium, or low? In other words, was there a good basis of randomized controlled trials for the intervention? Or in the case of some interventions, we're just looking effectively at observational data for some of the newer technologies. Get all the previous assessments that we've looked at and estimate the parameters associated with the model, the logistic regression, effectively predicting which technologies would we fund and which have we not funded. And that allows us to calculate an implicit threshold for different technologies and compare it to the stated threshold of 45,000. Well, in that model, if we have technologies that were classified as innovative and improve safety and tolerability, then we're willing to fund it at a higher level. Whereas if the quality of evidence was poor, even if it was cost saving, the decision maker didn't reimburse the technology. And that's called a revealed preference in economic terms. What does it look like in the cost effectiveness plane? Well, this is effectively what it's doing. So instead of having qualities on the x-axis, we now have a benefit function, which is a linear combination of parameters from the Logit model here. And that on the cost per quality framework, the drug may sit here, but on the benefit function, it sits further to the right. And so it gets reimbursed. Interestingly, couldn't we relax the assumption about the model that we're using? Well, that's the work that Shane was doing in Trinity in the area of MCDA. Question on tariffs and estimation of tariffs. Well, because of heterogeneity of the population, we fitted a latent class model to separate out subgroups within the population and estimate tariffs for those subgroups. And there's a method from management science, effectively, which tells us that perhaps we're good at choosing between two options, but we're not good about ordering all possible options. And so we can use the approach of pairwise comparisons of disease states. So even though someone tells you, oh, they're willing to trade off 10 years in that state for 1 year full quality of life, that sounds like a very big difference. Well, instead of encoding it like that, you could encode it and say, well, actually they prefer state A to state B. So if you look at pairwise comparisons, rather than looking at trying to elicit full time trade-offs, then maybe we can do something different. And that does change the tariffs slightly. So these are utility functions derived from pairwise comparisons rather than the standard time trade-off which is used within the EQ5D framework. The patients, again, was due to start this autumn, but the investigator has been on maternity leave. Second topic is about calibration. And here we've been working with Sheffield and the group in Harvard on models in colorectal cancer and in HPV, and recently in hepatitis C as well, which became a big topic in Ireland. It became a big topic in Ireland with the introduction of new drugs for the treatment of hep C. New expensive drugs, €1,000 per tablet, effectively, thereabouts, $1,000 per tablet. You have to take 80 tablets to clear hep C. I mean, we say cure hep C, but some people don't like that term cure. But so it's expensive, but actually a very cost-effective intervention. And we can demonstrate that it's cost-effective because you're preventing the need for a liver transplant down the line and the development of cancer, liver cancer. And so the calibration of these disease models is the topic, the second of these topics. And one of the examples is in the area of colorectal cancer, and there are two methods that have been used to calibrate this model. One is a simple, that's a Markov model, just demonstrating the model first, and the question is, can we estimate the transition rates between states? Now ethically, we couldn't investigate people and then find that they've got an adenoma and then just follow them up and then find, oh yeah, you've developed a high-risk adenoma, and then follow them up and find they've got stage A cancer and then follow them up. So you can't observe the transitions themselves, but you can observe cohorts of individuals of different ages, different numbers of people presenting with the disease. So you might observe some data, but not the transitions themselves. So given the information on numbers of cases in each of these states at different ages, can we infer something about the transition rates? Well, the idea is that we simulate values for the parameters and see if the predictions match the data. And when they do, that's a good fit. That's got high likelihood of those values of the parameters. So two approaches that we've taken. One is the Monte Carlo approach, simple Monte Carlo, which is a method that was used in a lot of groups previously. And the alternative is to use learning from the area of Bayesian inference, in fact, but as a tool of convenience rather than being fundamentally Bayesian, that you start somewhere and then you stochastically optimize samples from the parameter space. So it's the Metropolis-Hastings algorithm to try and find values for the parameter set that match our observed data. And you can see that the chains that are run, this is the sum squared error, so the overall fit to the targets over time. These are particular parameter values, and that it has this characteristic of converging to an area of good fit. And so we're sampling from collections of parameters that we can use for our probabilistic analysis, as was shown earlier. Here is an example of our targets. So this is the observed data, the black dots, and the grey values are values with high likelihood, if you like. So there's sets of parameters that fit the data reasonably well, and we use those for our model predictions. So that's the area of calibration. And the final area is that of evidence synthesis, which is a general term that we use for things like meta-analysis and collected similar approaches. Well, we'll talk about something called indirect comparison, and that's where you might have information from drug trials, but they compared, you don't have the explicit comparisons that you might want to know. So here are some drugs for rheumatoid arthritis, and what you really want to know is which is best, adalimumab or certolizumab, or adalimumab or golimumab. So you want to know which of these drugs is the best drug, but they haven't compared them head to head. But what you do have is a network of clinical trials given by the black lines there, adalimumab versus placebo or placebo plus methotrexate perhaps, adalimumab or golimumab versus placebo, collection of trials there, but you don't have them head to head. But we want to infer something about the indirect comparison of the agents? Well, let's have a look at this question. So let's say we have those two studies, drug A and drug B, drug A and drug C. And we have small studies, 12 people in each arm of the trials. And we find that drug B has 8 people having a good outcome. Drug C has 8 people having a good outcome. So the question is, which drug is better, drug B or drug C? Or are they the same? Now, this is a real question for a department like this. So if you see studies like this, what should we be thinking about? So we've got drug B in study 1 and drug C in study 2. And the same number of people, the same proportion of people do well on drug B and drug C. So can we class them as similar? Or what should we be thinking about? Well, we might be thinking there's different study populations. And how can we tell? Well, we do have a common comparator. So we've drug A used in both groups. And in study 1, 3 people do well on drug A. And in study 2, only 1 person does well on drug A. So that might indicate that the population in study 2 were sicker. So they were more frail, because only 1 person did well in study 2 on drug A, and 3 people did well on drug A in study 1. So actually, because study 2 group were sicker, and drug C had 8 people doing well, that's better than 8 people doing well in a population that weren't as sick. So it's the difference between drug B and A in each, and then drug C versus A, that we should be looking at, and that's called an adjusted comparison. So we do have some information that can allow us to indirectly compare between drug B and drug C in a setup like this. And this would be for randomized controlled trials, but then we have the question of, can we include observational evidence? And that's what's shown in the network there with the dashed lines, that we have observational studies comparing agents head to head, and a dotted line which is showing single arm evidence for a drug, etanercept, on its own, in fact, and a matched placebo arm that wasn't in the same study. So can we do that? Well, to answer the first of the questions, we can combine evidence from RCT data using the established approach of indirect comparison. So we do compare RCTs together, and we can get indirect comparison of AB from RCT evidence. We could do the same with observational studies, and we just label it as an observational comparison. But then to combine RCT data and observational data, can we get it at an overall level? And the answer is, we can adjust observational evidence in 2 ways. One is variance inflation, which says that the likelihood function from the observational study, we should just flatten it. A bit like saying we've got a confidence interval from our observational study, but actually we're not as confident with observational studies as RCT studies. So what do? We make it a bit bigger. We broaden the confidence interval. So we have variance inflation. We just say, let's discount the information. The other thing that we could do is we could shrink it to no effect. So we could say that the observational evidence, we believe it's overly optimistic, and we might shrink it back towards the null, if you like. And we do that using a couple of parameters. So the xi effectively is the weight associated. So that's the adjustment for variance inflation. And so if we set this parameter to 0, we have just RCT evidence, and then we increase it to 1, then we're treating observational evidence as if it was the same as RCT evidence. And we treat this as a sensitivity analysis rather than a canonical analysis. So we're saying, does it make a difference to the ordering of treatments if we include observational evidence as well as RCT evidence? And we're reassured if it doesn't make a difference. Now, it does make a difference to the effect, a bit of a difference to the effect, but if it's not making a difference to the ordering of treatments, it might reassure us that there isn't something strange going on. If it does change things, then do we believe the trials or do we believe the observational evidence? It was a conversation I had with someone who used to be in the cancer registry here, where we were discussing the impact of new agents in the area of cancer treatment. And we were looking at the experience in an observational cohort versus the experience in a clinical trial population. And I started the view saying, well, yes, so what we have to do is we have to adjust the observational evidence so that it matches the... And then I thought, and I said, well, no, hang on. The observational evidence is exactly what we see in practice, so that's what's important. And the clinical trial evidence may be in a situation where it works in theory, but actually it's what happens in practice that matters. So the impact of the adjustment should tell us something. So that's work that's already published. But the controversial thing is including single-arm evidence into a synthesis like this. If I just have a single observational study and I look at, let's say, the success rate of the drug, and then I match it to a study that I think is similar, then how good is the matching? What impact will that have? Can I just include it, do a matching, include it, and treat it as if it's an RCT? Or is it a really naughty thing to do? And we're told as statisticians it's a really naughty thing to do, but we're doing research, so we can do it anyway and see what happens. And it's early days yet, but the initial evidence is that if there is a large impact of covariates on the outcome, and there's a difference in the covariate pattern between the studies, then it's bad to include this single-arm evidence. In fact, it creates more noise than it does in helping you estimate the effect. It introduces extra heterogeneity where you don't want it. If you have large between-study variability, so you might see this if you're thinking about, say, a standard meta-analysis, and you see large heterogeneity between studies, that tells you large between-study variability, then it also turns out that it's a bad thing to do. But if the variability is low, then it's a good thing to do. And this is initial work, and it's ongoing work with one of the PhD students. So the main bit of work that we're engaged in is about what utility functions do we use, and we're looking at calibration of those across the population, calibration of models, disease models themselves, and then the question of evidence synthesis, and in particular, can we use this observational evidence? And we've more to do. Thanks very much. Is there any questions, I guess?
I guess I have two. One is just to what extent is this actually used in Ireland at the moment?
Is this framework?
Yeah.
Every new drug that comes to the HSE to be paid for goes through an evaluation of this type.
Right.
And the process is that the drug company, when it's applying for, it has license and its authorization to market its product. But if it wants to be paid for on the schemes, the GMS scheme and the drug payment scheme, then it has to apply to the CPU, which is the Corporate Pharmaceutical Unit of the HSE, for reimbursement. They come up with a price and they give evidence about how effective the drug is. Under legislation, the HSE has to provide essential medications effectively for the population. So if it's deemed that this is something the population needs, then the HSE will pay for it. Under the 2013 legislation, the HSE have to consider effectiveness, comparative effectiveness and cost effectiveness of the medications when they're making that decision.
So do they come up with a rule that says only prescribe it to certain groups?
Right. That's a very good. Okay. So that's a very good point. At the moment, the decision is to put it onto the schemes or not. And in principle, that allows for the situation where it could be used off license or prescribed for other indications than the indication that they're claiming reimbursement for. One of the big areas of work is to identify those subpopulations where the drug really works well, and in that case, the drug may be reimbursed in a slightly different fashion, and so that it made available to the population who would best benefit from it. And I think there was coverage in the papers at the weekend about a particular drug, which we'll hear more about, and some discussion being had with the company. Well, if the drug really works for these people, we'll pay for it. If it doesn't, we won't pay for it. And the company weren't willing to take the risk on themselves for whatever reason.
But exactly the decision process that the HSE go through at the moment, presumably, is not involves a lot of sort of input from experts in the field to medical understanding of disease and comparison outside of the country with different populations, and not just or utilities within the country. Is that right?
Right. So what happens is...
Tony's question was maybe, I thought was, to what extent is the utility component
from here
actually used in the decision process right now?
Yep. So what happens is that the company puts forward a bit like a document that's as big as a PhD thesis, if not twice as big, in fact. So they put forward a body of evidence that they're making claims for their drug, and they can use international evidence, and they use the clinical trial evidence, and they may use a pharmacoeconomic model that they've constructed or they've had a company construct to demonstrate the efficacy in terms of clinical efficacy of the drug, but the impact as well in terms of life years saved and life years gained, and the costs offset. So what costs am I preventing being spent in the system? And they make that application to the HSC, and that's reviewed by the centre, the National Centre for Pharmacoeconomics. In the case of some treatments, I should say that there's a clear case made on a very brief amount of documentation. But for often for high cost drugs or drugs that are in this top right quadrant, it's the big dossier that goes forward. Questions are then raised by the NCP and given back to the company, and the company have an opportunity to address those questions, and a summary report then is presented to the CPU, and there's a drugs committee which includes representatives from the HSC and people who have clinical background and so on, and they make a recommendation to the board of the HSC as to whether or not the drug should be purchased. But ultimately there have been situations where decisions have gone to cabinet when the HSC board has decided that it's not willing to fund an intervention or has referred the matter on. And it becomes a political decision then rather than a medical decision or a decision taken in-house. I should say that clinicians can, once a drug has marketing authorization and has a license, clinicians can prescribe the drug. It's a question of who pays for it in those circumstances. So the model that we have here is very much like the model that they have in the United States for a managed care system, where you might have certain medications that are reimbursed for certain indications and others that aren't. So our model is not different from anywhere else in the world, but perhaps what it looks like or the shape of it is slightly different.
And can I say, so the model that's used in Ireland, let's say used in the UK in terms of NICE and so on, so are they incorporating some of the Bayesian concepts?
Yeah. So the community, and I think we were just talking about the group. There's a group at Leicester that's very strong Bayesian group at the in medical statistics in Leicester, University of Leicester, and they've been quite heavily involved. Sheffield had a very strong Bayesian group. So the input from statisticians has often been in the Bayesian framework because the natural way in which we think of uncertainties, and also it clips well into the utilities and simulation type approach to the decision space. Yeah, so it is.
Because it's interesting, as you say, the actual medical literature is still dominated by the frequentist approach.
Yeah, I was going to say this, like I think cancer clinical trial organizations like the Southwest Oncology Group in the States, I think are averse
to the Bayesian.
To making decisions on the basis of Bayesian analysis, and I think substantially because the old criticism. Whose prior is it? I mean, okay, it's great calculation to make, but the question is that going to does that suit everybody? And it may suit a bunch of a specific subpopulation, or it may suit the sort of viewpoints of the analyst, but that may not be good enough. You know? So you need some kind of objectivity too. So
we very often in life and in science we like to think that there is such a thing as objectivity, but in any choices that we make, in statistics, when we make a choice about one model or another, there is a subjective nature to the decision and to the judgment. And what the Bayesian framework does is it puts it out there, and it admits that subjectivity. From a measure theoretic point of view, which is a slightly mathematical purist point of view, the classical framework often conditions on a point null hypothesis, so that conditioning on mu being equal to 0. If we want to create a probability measure on the parameter space, then we don't want to condition on a set of measure zero. So it's a finite point in a measurable space, and so we can't generate a measure on the parameter space unless we have a prior. So the prior is both open subjectivity, and it's a necessary evil, if you like, in a measure theoretic framework, that we need to have some prior. Where I would say that the Bayesian work has taken off is in the area of adaptive designs for clinical trials. So I know at the MD Anderson that there has been a growth in the Bayesian framework there, due largely to Don Berry, and I mean there were others there. Peter Muller was there, and a number of others, statisticians who would be come from the Bayesian way of training. I mean you're right that the frequentist framework is still very present in medicine, but we have had a couple of articles in the past, over the years, discussing the Bayesian framework. David Spiegelhalter, I know, and Palmer have published extensively in the medical literature as well. And within the statistics community, we've recently had an open discussion about the meaning of statistical significance and p-values. So the ASA has a statement on that area just published this year, which was written by both Bayesians and frequentists and objective Bayesians and subjective Bayesians. So you're right.
Well, I'm not challenging any of that, except that it still is the case. Okay, I mean, there's a lot of theoretical reasons why Bayesian analysis and decision making makes total sense, not that source. But then there is this point about this, a benefit of the approach is the subjectivity.
Yep.
But a main criticism of the approach
is the subjectivity.
I mean, and then I think that's been around for a long time.
But isn't it true that you can, isn't it true that you can fit different levels of subjectivity?
Yep.
You fit the best and the worst case.
Well, I mean, sure. But suppose you were a company and you were trying to make a case, and you knew that you had the opportunity to adjust the priors as well. Well, I bet you probably would.
Yeah.
Well, that's obvious. That's the obvious. I mean, that's an obvious fudge.
Yeah.
I mean, it's the stuff like changing the inclusion criteria in the trial, for example,
and there's a lot of sort of
that hides the
outstanding examples of observational studies that have gone awry for one way or other, and they're like inherent problems with that people, smart people will know about. But presume potentially if it was in the hands of a company, they might well decide that a particular study run at a very small hospital was suddenly the most important.
So there's two answers, and one is there is a framework called objective Bayes.
That sounds like a great term.
Yeah, where I mean, and a whole area of reference priors that are standard collection of priors that we use as the reference prior, if you like, which is neutral, that doesn't have subjective input. So that's one answer. And the other answer is to emphasize again that if I tell you what my prior is, you know where my subjective input is.
I'm just wondering, following on from that, has anybody ever done a study that looked at actually both approaches and see do you ultimately come to the same conclusion? Yeah. So for a lot of models, you can find a prior such that your inference will be the same as if you did a frequentist analysis. And do you end up with the same conclusion?
That you'd end up with the same conclusion. Yeah.
Just as a very nice case study in public health, people in the part will know, I've been talking about this for the last year or two, but there's been a series of studies published in JAMA and in the Lancet and in the New England Journal of Medicine, all from a small group of workers, suggesting that a low salt intake may actually increase your risk of cardiovascular disease and stroke. And this flies in the face of, say, 50 years of research on linking salt with high blood pressure and with cardiovascular disease, and it flies in the face of basic physiological mechanisms. So if you had any sort of Bayesian approach, the prior probability that this hypothesis was true would be very, very
small.
Low.
Yeah.
But yet these studies have been presented, and the methods are quite dodgy. They're each dodgy in a slightly different way. So that's. but they're still presented as if nothing had ever been done on this
Yeah,
topic
yeah.
before. And they've had a huge impact, you know, they've had massive uptake in the media, here in this country. The Irish Heart Foundation want to convene a group looking to actually change our current
Guidelines.
just based on these studies. Whereas, in a way, it would be very nice
Yeah.
case study for somebody just to take those papers and maybe put them through a Bayesian type
Yeah.
analysis.
So that's I mean, we think of it in terms of sequential meta-analysis would be a traditional way of thinking of the Bayesian paradigm. So that if I think about going back in time for all those studies, go back to the first study, well, I might have had a very flat prior to begin with, and I include the information from that study, and now I've got a distribution. And then I add in the next study that happened in time, and so on. So that's like a cumulative meta-analysis over time. And gradually what happens is the distribution gets narrower and narrower and narrower, but it also means that if a new study flies along that's completely different, it doesn't move the body of evidence too much. And that's the way it should be. So that we have this inertia, that we don't just jump to the next thing.
It's interesting, is that for most people in this field, their belief hasn't shifted a jot.
But the media,
for the people who actually have published the work, obviously they're completely convinced.
Yeah.
So you have these two groups that are utterly polarized.
and but also that's the way that in some ways, that's the way I think it should be, is that if we're exposed to the same information, we can agree. But if we haven't been exposed to the same information, we're allowed to disagree. And gradually over time, when we're exposed to enough science, we converge. So if we have a common information set, if it's sufficiently robust, we'll agree eventually. We'll come close enough. We might be perfectly in agreement. Distributions aren't exactly the same, but they become close. And when we get to somewhere when we're close to each other, then we can stop fighting about it.
Well, that never happened.
How much consensus regulation? I mean, I'm just thinking this might be another area that maybe the Bayesian stuff would really help us. Happens in improving drugs. To some extent, like you take an issue like global warming. Basically, individual groups can sort of have their own opinion, but in the end, it seems to have sort of evolved towards having a sort of a consensus view that provides a mechanism for different groups to pool data so that they come up with the same or come up with
similar
a agreed position. So, does that happen with regulation of drugs in Europe?
Well, I mean, in fact, I mean, there are a number of different ways in which we can have that a drug can be regulated, and if when it's done centrally, that's one story. But it is possible for a drug to go for approval in a member state and then be approved Europe-wide on the basis of effectively the decision of one member state. And the question then is, do we get consistency in how drugs are approved? And that's a different day's conversation.
I imagine that would be pretty useful. I mean, because Ireland is such a small group, companies can't be particularly interested in
this market.
Yes.
Okay, I'd like to thank Cathal and for a great talk and even more robust discussion.
Thanks very much.